So we'll go into the next part of the same class. This is in continuation with the previous class. Okay, today we will learn about amino acids and their properties. What is a Zwitterion? Very, very important topic because it comes as a short note and even in Viva. Then we have colored reactions, which helps in detection of amino acids. There are certain derived amino acids, which are not exactly the 20 amino acids. There are certain amino acids which are derived from these 20 amino acids, and what are they and their importance. And there are special functions of amino acids beyond just forming protein. Okay, so we'll learn about that. Let's go one by one. So we have properties, physical and chemical properties. First, we will go through physical property. Solubility. They are soluble in water and alcohol, which are called polar solvents, and they are insoluble in non-polar solvents like benzene. Melting point. They have a very high melting point. Usually, melting point is above 200 degrees Celsius. Now, some of the amino acids exhibit some taste in them. Okay, some of them are sweet, like glycine, alanine, valine, etc. There are more. Some of them are tasteless, like leucine, and some of them are very bitter, like arginine and isoleucine. Optical properties we have already read before, right? The optical isomerism because of the presence of anybody? Asymmetric carbon. Okay, very good. And all of the amino acids show optical isomerism except glycine. Very good. Okay, so optical property ho gaya. We have something called Ampholytes. So amino acids have, as you remember, if you remember from the structure of amino acid, it has carboxyl group and it has amino group. So it has both positive charge, amino group में positive charge होता है and carboxyl group में negative charge होता है. So any molecule that carries both the charges, they are known as Ampholytes because they can accept a proton as well as donate a proton. Okay? So they act as an ampholyte. Now what is this? Come on, you know it. What is the basic bazaar wala name? Sugar free, right? Or sugar free gold. What are these actually? Artificial sweetener. Okay, and given to which people? Diabetic people and those who are fitness conscious, right? Now, some of them they use stevia, some of them they use aspartame, some of them use sucralose. Very good. So these are just, they are not glucose, they are not sucrose, the table sugar. So except that table sugar, what we have is amino acid as well. Here, aspartame is used in many of these sugar-free gold, right? Aspartame is made up of amino acid called aspartic acid and phenylalanine. Okay, so like I said, it has some of them have sweet taste. So aspartic acid and phenylalanine being one of them, so they are used in place of artificial sweeteners so that it doesn't affect it is not a carbohydrate and it doesn't affect diabetic people. All right. What is this? Chow mein. Okay. Which kind of food is this? Chinese food, right? So you're getting what I'm saying. What makes, how many of you like Chinese food? Almost all of you, right? What makes Chinese food so tasty? What is MSG? Very good, monosodium glutamate, right? So this is what I'm getting at. Monosodium glutamate is a derived form of glutamate, okay? And this is an amino acid that is flavor, it enhances flavor. So it has an flavor enhancing property due to which it is used in food industry, not only in Chinese food, in all kinds of food it is used, all right, to enhance the taste and give it a flavor. Now some of the people who have, there are cases where people have gone to the restaurant or brought in Chinese food and they, after having food, they experienced lightheadedness, dizziness, breathlessness, headache and all of those things and flu-like symptoms. That has been linked to the presence of monosodium glutamate in food. Alright, so this is known as Chinese restaurant syndrome. Although it is only transient, it is not a disease, it just happens for a few minutes and it goes away. But this has been debunked many times, it has been outdated. Long few decades ago it was still there, where people used to just avoid MSG because of these symptoms, but recent studies have found no link that correlates between the use of MSG and the presence of these symptoms. Okay, but this is just an extra knowledge for you. Now, another property of amino acid is absorption. So there are certain wavelengths at which it absorbs the amino acids in the solution, absorbs the UV light or absorbs the light in the visible spectrum. And tryptophan is one of them. It has the maximum absorption capacity at 280 nanometer. Okay, so if we want to find out the presence of tryptophan in a solution, we will fix our machine that detects the presence of amino acid at 280 nanometer. Okay, you will not understand this as of now. We will do experiments on it and then you will understand it later and we'll explain to you later on. But for now, just remember tryptophan and 280 nanometer wavelength. Alright. Then we have something called Zwitterion. What is a Zwitterion? A Zwitterion is any hybrid molecule that has both positive and negative ionic groups. You know, amino acids have both positive and negative ionic groups in them. Positive being the amino group and negative being the carboxyl group, right? But what is the property of Zwitterion? It has no net charge. Even though it has positive and negative charge, it is equal number of them. So see, every amino acid is not a Zwitterion. There is a particular pH at which an amino acid starts to behave like a Zwitterion, and that particular pH at which a Zwitterion, an amino acid acts as a Zwitterion is called isoelectric point or isoelectric pH. Okay, so although it has both positive and negative ionic groups in them, they are equal in number when acting as a Zwitterion. So Zwitterion का क्या property है? They have both positive and negative charges in them, but since they are equal in numbers, they cancel each other, so they are electrically neutral. So their net charge is 0. So what happens? What is the point of learning Zwitterion? Zwitterions have certain properties. They are not soluble. They have the least solubility, okay? And they are precipitated easily at their particular PI, at their particular pH. तो हर एक amino acid के पास एक ऐसा particular pH होगा जिसमें we can precipitate them. Precipitation means we can separate them from the solution. Since they are least soluble, they form clumps. You will see this in when we do protein experiments on protein as well. They form clumps and they solidify and we can easily take them out. Alright, so that happens at a particular pH for each amino acid. हर एक amino acid का PI different है, and at those particular pH at which the amino acid acts as a Zwitterion is known as isoelectric pH or isoelectric point or PI. At that PI, the amino acid can also act as a buffer. What is a buffer? A buffer is any substance that will resist any change in the pH of the body. So this helps in maintaining pH of the body. pH क्या होता है, कैसे maintain होता है, and all of that we will learn later on. But just remember this point, alright? Then we have the point that Zwitterions do not move in any electric field. I don't know in school if you have done electrophoresis. I don't think so. Okay, so electrophoresis is a method in which we separate certain molecules in the solution, say DNA or any positive or negatively charged molecules. So we put them through an electric field, and the ones that are positive in charge, say DNA is negatively charged, so it will move towards the positive pole, and the molecules that are positively charged, they'll move towards the negative pole because of their polarity, because of the charges in them. But क्योंकि Zwitterion के पास कोई charge नहीं है, net charge is not there, it is 0, it doesn't move in the electric field. Alright? So see over here, you see this is basic, any standard amino acid, right? This is in free form, in cation form, in acidic pH, low pH means acidic pH, इसमें they act as cations, which means they're positively charged. In alkaline pH they act as negatively charged. Okay, and Zwitterion में देखो they have both positive and negative charge. All right, this is called the Zwitterion. It is called a Zwitterion. Okay, this comes as your viva as well as short note. So please remember Zwitterion. The definition of Zwitterion should be in your fingertips. We have learned about physical property. Let's talk about chemical properties as well. Now, amino acids form bases, salts with bases, and esters with alcohol. Whenever an amino acid will react with alcohol, it will form esters. This we will see in fatty acid synthesis as well. And with bases, say for example, sodium hydroxide, it will form salt. Okay. Then a property called decarboxylation. From the name itself, you can make out decarboxylation means the removal of the carbon dioxide. Okay, it can undergo decarboxylation and form amines. Over here, ये चला गया. Okay, and बचा क्या? NH3. The amino group remains. So this is called an amine of an amino acid. Okay. So they are histamine. नाम से पता चल जाता है histamine from histidine. Okay, histidine हमारा proper amino acid है, उससे हमको मिला histamine through decarboxylation process. Okay, same with tyramine from tyrosine. Very good. And GABA or gamma amino butyric acid from glutamate. Okay, not glutamine. Glutamine is an amide. Okay, please remember this, don't forget. Histamine, tyramine and GABA, very important products of decarboxylation. We will learn about them in the next few slides as well. Okay. Then they react with ammonia. So this is because of the presence of carboxyl group. Okay, carboxyl group ka reactions hai. Then we have reaction with ammonia. So this is the amino group. Whenever an amino acid reacts with ammonia, it forms a corresponding amide. Okay, not amine, amide. So aspartic acid reacting with NH3 will form asparagine. Glutamic acid will form glutamine. Glutamine is not an amine, it is an amide. Please remember this, okay? And the amino groups behave as bases and be combined with acids to form salts. Then we have something called peptide bond formation. This we will keep on repeating throughout biochemistry. What is a peptide bond? Alpha carboxyl group of one amino acid reacts with alpha amino group of another amino acid to form a peptide bond. So you see here CONH. This is a peptide bond. A peptide bond is formed between two amino acids. दो amino acid जुड़ के एक जब जुड़ते हैं तो they make a peptide bond. What is the bond in carbohydrates? Glycosidic bond, right? And in fats it is called ester linkage. The same way in amino acids we have peptide bond. But that bond is not formed just randomly with anybody, okay? Alpha carboxyl group. What is alpha carboxyl group? The asymmetric carbon के साथ जो carboxyl group जुड़ता है, that is called alpha carboxyl, and same with the amino group that is joined to the asymmetric carbon. उन दोनों ने मिलके over here you see, this is the asymmetric carbon. You have carboxyl group here, so this is alpha carboxyl, this is alpha amino. दोनों ने मिलके ये बनाया. This is called a peptide bond, and this is called a dipeptide. So dipeptide doesn't mean two peptide bonds. It means one peptide bond between two amino acids. Okay. Peptide bonds will cause, so there will be chain of amino acids. They will be all joined together by a peptide bond, and the whole chain will be known as polypeptide. Okay, and chains of polypeptides will form a protein. Next we have colored reactions. So certain amino acids have on reaction with certain substances form colored reactions. So what's the point of learning this? This helps us in separating certain amino acids or isolating them from the solution. Alright, so अगर किसी चीज़ को हमको पता करना है for a particular amino acid, we know that this amino acid is supposed to form this color with this substance, so that will help us in the detection of that amino acid. One is called ninhydrin. So the D-amino acids, we know all the amino acids, most of the amino acids are in L form, but the D form of amino acids is what we are talking about here. So they react with ninhydrin to form a purple, blue and pink color complex, which is called as Ruhmann's purple. So this is purple. अगर उसमें हमको D amino acid नहीं मिला, then this will have no color. But when there is a presence of D amino acid, then we will find that it has turned to purple. Another thing to see here is that proline and hydroxyproline also give a color with the same ninhydrin, but a different color, which is yellow in color. Okay. It helps us in determination of D-amino acids. It helps us in determination of the presence of proline and hydroxyproline amino acid. There are many colored reactions. I'm not going to talk about it right now in the class. It's there in your books. Remember them. But one important colored reaction is biurate reaction. The principle behind it is, I'm not going to talk about it because we'll have classes specifically for it. Biurate reaction usually we do for the presence of dipeptide. Peptide bonds जहाँ present होते हैं उसको देखने के लिए, or the presence of proteins. That will cause a presence of, that will cause reaction, just purple in color. Okay, so this is purple colored reaction. Biurate test is positive, so it will show purple colored reaction. Okay, biurate reagent in itself is blue colored. So if blue turns purple, then we know that there is presence of protein in them or certain amino acids in them. Similarly, for the presence of SH group, which amino acid has SH group in them? Sulfur group in them. Anybody remember? 2 amino acids that has sulfur group in them. Yes. Very good. Cysteine. And anything else from M? The letter M? Methionine. Cysteine and methionine. So this is specifically for SH group, which is in cysteine though. So this will form deep red color or purple colored reaction with the same substance known as ninhydrin. Okay. Another important property, chemical property of amino acid is transamination reaction, and you will learn about it again and again throughout the whole course of biochemistry. Transamination, नाम से पता चलता है. Trans means transfer, amine, transfer of amino group. Okay, from an amino acid to a keto acid to form a new amino acid and a new keto acid. Okay, this is glutamate, this is pyruvate. So this is glutamate is an amino acid. Okay, इसका जो amine group है, amino group है, that gets transferred to a keto acid. So this is carbonyl group. This is a keto acid. Okay, pyruvate is a keto acid. So this will get transferred here and form this alpha keto glutarate. तो अब ये वाला जो है इसकी जगह हमको ये मिला. Okay, this is replaced by this part here over here. So it becomes, this is now has become keto acid. Any amino acid that has this group is known as keto acid. Okay, so it has, the original amino acid has become a keto acid because of the transfer of the amino group, and the original keto acid has become an amino acid over here known as alanine. Alanine, you know, structure change हो गया, it has converted into an alanine. So this process is known as transamination reaction. Transfer of amino group from one amino acid to a keto group, keto acid, so that the original amino acid becomes a keto acid and the original keto acid becomes an amino acid. Okay, this is through the enzyme alanine aminotransferase. It's very important. You will learn about this in liver function test as well. The importance of it you will learn later. Remember the name. Same with transamination, we have oxidative or deamination as well. Deamination. So the amino group over here, the amino acids undergo oxidative deamination to liberate free ammonia. Glutamate is an amino acid. So the amine group over here gets removed, deamination, removal of amine group, okay? Through the enzyme glutamate dehydrogenase, ये बीच में एक byproduct बनाता है, and then ultimately you form alpha keto glutarate, which is a keto acid plus the liberation of ammonia. Okay, amino group is released as ammonia in this process through oxidative deamination. So now we have talked about the properties. Now we'll talk about derived amino acids. So you understand there are 20 amino acids. In nature there are 300 amino acids, but in human body we are concerned only with 20 amino acids. But besides those 20 amino acids, there are several other amino acids which are biologically important. Okay? And after the formation of these amino acids and proteins, some of them undergo post-translational modification, which means that amino acid together formed protein through the process of translation, and after they have become protein, they undergo modifications in them so that it becomes fully functional or get activated. So some of them are collagen, which is the most abundant protein in mammals, and it contains hydroxyproline and hydroxylysine. You will learn about them in the vitamin chapter, also in the chapter of scurvy as well. You hear about this collagen in many creams and lotions and in the beauty industry as well, right? So this is collagen, which is a derivative of of these amino acids. It is there. It helps with the elasticity of the skin, with gums, and it has huge role in the body. We will learn it later on. Then we have something called histones. So histones are those circular molecules around which the DNA gets winded up. It gets circled around those proteins. So they are called histones. Those are also made up of certain amino acids. Then we have gamma-carboxyglutamic acid. They are found in the process of blood clotting. So clotting of our blood also has these amino acids. Certain amino acids are involved in there as well, known as gamma-carboxyglutamic acid. Then we know cysteine is there from cysteine and cysteine condensation. D-amino acid, we know all the amino acids are of L category, but then there are certain amino acids of D category as well. So they are found in certain antibiotics like actinomycin D, valinomycin, gramcidin S. They are antibacterials. All right. D-serine and D-aspartate are found in brain tissue. D-glutamic acid and D-alanine are present in certain bacterial walls. Okay. Now finally, some special function of amino acids. I said in the previous class also, all the amino acids, all the proteins are formed by amino acids, but not all the amino acids form proteins. Okay, amino acids have functions beyond formation of protein as well, as we have seen already. Then there are certain important functions of amino acids beside forming proteins. For example, we have gamma-aminobutyric acid, I said before also, GABA, and dopamine. So GABA is derived from glutamic acid and dopamine from tyrosine. Okay, they act as neurotransmitters in the body. Neurotransmitters means they help in transmission of neural circuit. Okay, signal from one brain tissue to the body or from one brain cell to the other brain cell. All right. Histamine from histidine, right? And amine from histidine is a mediator of allergic reaction. Anything that involves an allergy in the body, be it the bee sting, be it any food allergy, whatever it may be, say asthma, histamine is involved. You will learn later on in your pharmacology, also in your second year, that all the anti-allergics that we take, they are known as antihistamines. Okay? Kuch bhi, for whatever reaction you may have in the body, the doctor will prescribe you antihistaminics, which block these histamine release from the body, because histamine is involved in allergic reaction in the body. Okay, this is also an amino acid. Thyroxine from tyrosine is a thyroid hormone. We all have thyroid glands in our body. From there, thyroxine is released. Okay, this is a thyroid hormone. Serine forms the active center of many proteolytic enzymes. So what is a proteolytic enzyme? Proteolytic enzymes are the enzymes that breaks down substances, that breaks down protein. Okay, उसके अंदर में भी serine रहता है. So these are certain amino acids which are not only involved in protein formation, but they have a variety of other functions as well as described. We heard about histidine as well. They are important in the buffering activity of protein. Alright, we have lysine, we have ornithine and citrulline. They are derivative of arginine and essential for urea synthesis. So you will learn urea cycle later on. Urea cycle mein kya hota hai? You know, ammonia is toxic for the body, especially for the brain. So our body forms ammonia, okay? But ammonia cannot be circulated in the body because it is toxic. So we need to remove ammonia and then convert it into a milder form, a non-toxic form. That non-toxic form is called urea. So uska pura cycle chalta hai, okay? That is called urea cycle. Through the process of urea cycle, we detoxify ammonia in our body. And for that, we need ornithine and citrulline. You will, in the urea cycle, you will read about it. These are also certain derivative of amino acids which are used not in the formation of protein but in urea synthesis. Alright? Now, potentially toxic amino acids. There are certain amino acids which can be toxic as well. They are present in plants. Alright? So, kesari dal. Have you heard of kesari dal? Anybody? So this, I think the government of India banned it a long time ago, the growth of cultivation of kesari dal. So this is how kesari dal looks like. It may look like something else also. So kesari dal or the Lethyrus seeds, okay. So they have something called beta or DAP or beta oxalyl diaminopropionic acid. So the seeds of this dal has this particular component in them which causes lethyrism or letharism. So what happens in letharism? There is a paralysis-like state in the body. Okay, you can say paralysis only. There is muscle atrophy, which means the muscle starts to break down. Okay, so muscle wasting is there. There is hypotonia, people stop working, like you cannot walk anymore, you have paralysis. Then neurological symptoms also appear because of the consumption of this dal. Okay, so not just when you take the dal, in moderate condition it's fine, but if we are completely dependent on taking Lethyrus seeds or this particular dal, that's when we have these symptoms. Alright, but there are studies now that say that there isn't much, this is safe to take in moderate amount. This study was done in a particular time where people were going through starvation, and this dal was the only staple food they had, and they probably had taken it in a very high amount, due to which many of them had paralysis. Okay, so neurological symptoms, and that's why government banned it. Cultivation still goes on, but this is just a side note. Okay, so this is called lethyrism. So this Lethyrus seeds also has alpha and gamma diaminobutyric acid, which also disrupts urea cycle. So जब urea cycle disrupt होगा, what will be produced? Ammonia will be produced. Okay, urea will not be synthesized. Ammonia will not get converted into urea, and then we'll have toxic ammonia circulating in the body. Ammonia, when it enters the brain, it will cause neurological symptoms. Okay, so that's why we see so many of these symptoms in these patients who consume kesari dal. Then we have something called cycad seeds. These are the seeds of that particular plant, which are neurotoxic, and they're implicated in Parkinson's dementia and ALS. What is ALS? Amyotrophic lateral sclerosis. Any important person that you know who had ALS? Who's this? Stephen Hawking, right? He passed away, but he suffered from ALS his whole life. So he is an exceptional case of ALS. Usually people who suffer from ALS, they don't have a very good prognosis. Within a year or maximum of 15 months after the diagnosis, they're supposed to pass away. But this guy lived up to, I guess, 53 years or something. So that was an exceptional case. But obviously, quality of life was not there. So in cases of Parkinson's dementia as well as ALS, we see the presence of, we see the role of these seeds as well. So patients or anybody who takes seeds from these plants, they have these kind of symptoms. All right. So this was short class for today.